Okay, hello friends. Again continuing with the photosynthetic pigments. Last time we discussed chlorophyll, we discussed different types of chlorophyll. So today I am starting with carotenoids. Carotenoids and phycobilins is my aim for today's session. Very quickly finishing off with carotenoids. First of all, you should know carotenoids, they are yellow-orange in color. So they will impart color starting from yellow till orangish shade. Okay, so yellow, it could be yellow, it could be orange, it could be yellowish orange. Okay. Secondly, that you should know that carotenoids, they are going to be lipid soluble. You can very easily dissolve these pigments in lipid. Talking about the structure of carotenoids, the structure of carotenoid is terpene structure. It is made up of 4 terpene structure. 4 terpene structure. So you can say tetraterpene in nature. Tetraterpene. Okay. And each terpene, when we are talking about terpene, so if I am talking about just one terpene, so each terpene will have 10 carbon atoms in it. 10 carbon atoms. So a total of how many carbon would be there in this tetraterpene? One terpene has got 10 carbons, so 4 terpene will have 40 carbons in it. Okay, so this carotenoid has got 40 carbon in it. Okay, the end of this carbon chain, this can be open or this can be closed. End of this terpene is the ring, na? Carbon ring. So end of this carbon ring, or not the ring, but you can say chain. Not the ring, we will call it as chain. Carbon chain. It can either be open or it can either be closed. Can be open, can be closed. Okay. So when it is going to be open or when it is going to be closed, if a cyclic structure is absent, okay, if there is no cyclic structure at the end, then you call them as to be open. If cyclic structure is absent, then you call it as open. But when you're going to call it as closed, if at the end of the chain there is a cyclic structure present. If cyclic structure is present, then you call it as closed chain. Okay. Talking about the types of carotenoids. Types of carotenoids. So there are 2 main or broad classification of carotenoids. They are divided into 2 types. First is carotene. Another one is xanthophyll. Now always remember there are 2 terms used in biology. One is phyll and other one is phobic. If you are calling it as phyll, it means loving. If you are calling it as phobic, phobic, P-H-O-B-I-C, if you are calling it as phobic, at the end that means it is scaring. It is getting away. It is repulsion. There is repulsion in it. Okay. Coming over to the carotene. Now the formula for carotene stands to be C40HX. So it will have a fixed 40 number of carbon atoms in it along with hydrogen. It's a hydrocarbon which does not consist of oxygen. So in carotene there would be no oxygen. Biggest difference between carotene and xanthophyll. Xanthophyll will contain oxygen in it. Contains oxygen in it. This is also a hydrocarbon having carbon, hydrogen, but along with this it will also have number of oxygen atoms. Okay. So over here I told you the formula for carotene. Same way, xanthophylls, you can say they are the oxygenated forms of carotenes or carotenoids. These are oxygenated forms of carotenoids. So they are C40H56 mostly. Oxygen will be X. Now this X will vary from 1 to 8. Okay. This X over here in this H is usually 56. Okay. Now coming over to the color, carotene is usually orange to orangish red in color. And xanthophylls, when you talk about this one, this is going to be yellow in color. Carotene, it gets completely dissolved in carbon disulfide, CS2. So carotene is totally soluble in this CS2. But when you talk about the xanthophylls, they only get partially soluble. partially soluble in CS2, carbon disulfide. Okay, the most abundant carotene is beta carotene. There are several types of carotene. I'll give you the examples. Beta carotene is in the majority, it is the most abundant carotene which is found, and this beta carotene, it is going to show absorption of light at the wavelength of 400 nanometer to 500 nanometer. Okay, absorption spectrum. Absorption spectrum means it's going to absorb light at 400 nanometer till 500 nanometer. Okay, and same way here, if you talk about the xanthophylls, absorption of light would be slightly either below this range or above this range in case of xanthophyll. Okay, slightly below or slightly above 400 nanometer. Example of carotenes can be, I gave you, beta carotene could be there, alpha carotene, gamma carotene are there. Then we have got lycopenes. Lycopene is usually present in tomato. One thing you should remember, beta carotene is very, very important. It is required in human body, or even in case of the plant body, for the formation of vitamin A. Okay, for the formation of vitamin A. Why? Because this beta carotene has got a ring which is called as inon ring. Okay, and any substance which have got that inon ring, I will tell you, inon ring. Okay, any substance, if it has got an inon ring, then that will possess the ability to form vitamin A. Vitamin A synthetic activity would be there. And this beta carotene is actually responsible for imparting orangish to reddish color to the reddish. Okay, then same way, coming over to the example for xanthophylls, that is lutein. Lutein could be there. Then there could be zeaxanthin. Zeaxanthin. Then there can be neoxanthin. There could be flavoxanthin. Okay, all these things. And then there could be alpha, cryptoxanthin, and beta cryptoxanthin. Now, in case of these xanthophylls, the only xanthophyll which will possess an inon ring is going to be the beta cryptoxanthin. So, this beta cryptoxanthin will have the ability to form vitamin A. Okay. Usually in case of carotenes, beta carotene, alpha carotene, gamma carotene, all these three of them will possess this inon ring. So, they will have the ability to synthesize vitamin A. But alpha and gamma possesses inon ring in a very few quantity. So, they can help in it, but alone cannot result in the formation of vitamin A. So, up till this point, it should be clear to you what exactly are carotenoids, what are the types of carotenoids, and this is all they would be asking to you, not more than that. Coming over to the functions. Functions of carotenoids. Very, very, very important. Functions of carotenoids. There are two main functions of carotenoids. First is, it is going to act as a helping pigment, or it is going to act as the accessory pigment. Secondly, it will act as a shield pigment, protective pigment. So, it is helping pigment and it is acting as the protective pigment or shield pigment. So, it will be acting as a shield from what? Shield from photooxidation. It protects chlorophyll, any chlorophyll A, B or C, any chlorophyll from photooxidation. Okay. Now let us discuss these 2 points one by one, accessory pigment and shield pigment. When you are talking about the accessory pigments, you know main pigment in photosynthesis is chlorophyll A, the primary photosynthetic pigment, which actually absorb the light energy and change it into the chemical energy. Primary photosynthetic pigment, that is chlorophyll A. So, a chlorophyll A, you know what, each and every pigment has got a very narrow spectrum, has got a very narrow absorption spectrum. It can absorb the light only in a specific wavelength. Okay, so if we are talking only about chlorophyll A cannot absorb a huge amount of light energy alone. It will absorb the light in a very narrow range of wavelength. Okay, it only absorbs light energy from the blue violet region and red region. Always remember this thing. Chlorophyll absorb it either from blue or it absorbs from the red region, violet blue or red region, from these two regions. What about the other regions? From there also the sunlight has to enter. So that function is done by these carotenoids. Carotenoids can help if they can absorb light energy of wavelength other than that absorbed by the chlorophyll. Okay, and then once they get these wavelength, once they get the light from these wavelength from other than the absorbed by chlorophyll A or chlorophyll B, then after that once they absorb it, then they will transfer this light energy to the chlorophyll A. So main would be chlorophyll A, who is actually performing the function of conversion. But these carotenoids, or even if we talk about the xanthophylls, what they are doing, or even if we are talking about the phycobilins, what they are doing, they are simply receiving the light from other areas and they are sending that light to the chlorophyll A so that it can convert those light into the chemical energy. Okay, so by this carotenoids are increasing the absorption spectrum of chlorophyll A. That is the one function of accessory pigments. Second function is the shield pigment. Now this is something which you really need to understand what is happening. See, whenever light energy is high, whenever the light energy is too much, this high level of light energy, they actually ask the chlorophyll, or they activate the chlorophyll, and chlorophyll undergoes oxidation. Okay, chlorophyll will undergo oxidation. That means this activated chlorophyll, it can react with the oxygen radicals. Okay, this chlorophyll will react with the oxygen radical, which can cause the destruction of chlorophyll structure. Destruction of chlorophyll structure. So are you understanding what do you mean by photooxidation? Photooxidation means oxidation of chlorophyll is taking place in high light intensity, in high light energy. Okay, so whenever a light intensity is higher, whenever there is more amount of light energy, it is going to destroy the chlorophyll slowly, because in that case chlorophyll starts reacting with oxygen radicals, which results in eventually destruction of chlorophyll structure. Okay, now these carotenoids which are there, they will protect this chlorophyll A from being photooxidized. Basically we are talking about chlorophyll A here. Okay, these carotenoids, they protect chlorophyll A from being photooxidized, from getting photooxidation. How they are protecting? See, in carotenoids at end there can be, I told you that there is either the carotenoids chain is going to be open or it is going to be closed. If it has got a cyclic structure, it is going to be closed. So sometimes in carotenoids at the end there is presence of a cyclic group. Let us say this is the cyclic group. Okay, so there is a closed structure present. This is that structure. Okay, now these closed cyclic group, they can very easily react with the oxygen radical. Okay, this is the cyclic group. Now this cyclic group will start reacting with an oxygen radical, and as soon as they react with the oxygen radical, this covalent bond over here, it is going to break in the cyclic group and it results in the attachment of oxygen. How? See, like this. So this covalent bond which was there, it is going to break and it results in the attachment of oxygen with this cyclic group. Now this carotenoid, so this we are talking about the carotenoid, isn't it? So this was the chain which was having the cyclic structure at the end. So this carotenoid now, after being attached with the oxygen, this is now going to be called as epoxy carotenoid. Okay, initially this was non-epoxy form. Okay, this was non-epoxy form of carotenoid. Now if oxygen radical is reacting with this cyclic structure of carotenoid, then there would be no more free oxygen radical. Any oxygen radical would immediately react with the carotenoid. So now no more oxygen radicals would be free, so that they can cause the oxidation of chlorophyll A. It's the oxygen radical which is causing the oxidation. Now if they are not present freely, so they cannot let the chlorophyll A get photo-oxidized. So in this way, carotenoids are getting converted into epoxy carotenoid and thus helping chlorophyll A to proceed normally. This is how they are going to protect the chlorophyll. Okay. Now, if the oxygen radicals were free, then they would have reacted with chlorophyll A, but now carotenoids have reacted with those oxygen radicals to protect the chlorophyll. Okay. Now, this is what is happening during the daytime when you have the light intensity. But during the night time, this epoxy carotenoid will again get converted back to non-epoxy form. This is during night time. This is during the night time and this is during the daytime. Okay. So epoxy form gets converted to non-epoxy form using an enzyme, that is enzyme D-epoxidase. This is the enzyme D-epoxidase. So always remember the two functions. This shield pigment, carotenoids, they act as the shield pigment, or how they are protecting chlorophyll A from being photo-oxidized, or how they are helping chlorophyll A, or why they are acting as the shield pigment. These are very frequently or very nicely asked questions in the medical entrance examination. So please focus on this portion. Next comes the phycobilin. After carotenoids, the next pigment that we have is the phycobilins. Now phycobilins, you already know it is present in red algae, isn't it? So red algae will have chlorophyll A, chlorophyll B and phycobilins. Now phycobilins, I told you, they are of three types. There is phycocyanin, then there is allophycocyanin, and then there is the third one, which is phycoerythrin. These two, they are blue in color and this one is red in color. Okay. In red algae, when I am saying red algae, I told you that these phycobilins, they are present in red algae. But now this algae is having phycobilins. It is also having phycocyanin, allophycocyanin and phycoerythrin. All these three pigments are there in red algae. But why then it is appearing red in color? Why not blue? The reason is that concentration of phycoerythrin is in very, very high amount as compared to phycocyanin or allophycocyanin. That is why it is appearing red in color. Talking about the structure of phycobilins, it has got 4 pyrrole rings. Again, same as that of the chlorophyll, they have got 4 pyrrole rings. But now this pyrrole ring, they are connected in such a manner that they are making a chain-like structure, like this. So now they are attached in a manner that they are making a chain-like structure. In chlorophyll A, they were a closed tetrapyrrole structure, but here it is open. Okay. In this case, tail would be absent. Magnesium is absent. This is water soluble phycobilin. They are water soluble and they are heat labile. Okay. They are heat labile. Then basically phycobilin, when you are talking about the phycobilin they joins with protein. They are going to show the association with specific proteins and they will form phycobiliproteins. Okay, they will form phycobiliproteins. What are phycobiliproteins? Now these phycobiliproteins, they will aggregate, they will join to one another and they form a group over the thylakoid membrane. Let me show you. Let's say this is the thylakoid. I am showing, this is not chloroplast, I am showing you this to be thylakoid and this is the thylakoid membrane. Okay. Now phycobiliproteins, they are going to get aggregated. Let us say this phycobilin is joined with protein, so they form this phycobiliproteins like this. Okay, now these phycobiliproteins, they will start joining with each other. They will aggregate together. They form a group one over the other and this will form a complex-like structure. This is forming a complex-like structure which is called as phycobilisome. Phycobilisome. Always remember. Okay. So they will form in red algae, they will get aggregated. Phycobilins, they join with protein over the thylakoid membrane and they form a complex-like structure which is called as phycobilism. Function of phycobilins, again they act as the accessory pigments for absorbing light energy and transferring it to chlorophyll A. Same thing, they act as the accessory pigments. These are the functions. Then second was, they are associated with one very important phenomenon which is called as Gaidukov phenomenon. Also called as complementary chromatic adaptation. Complementary chromatic adaptation. Okay, now this we are going to be discussing this exact phenomenon, Gaidukov phenomenon. This is a topic of bacteria. These Gaidukov phenomenon, it is shown by specifically blue-green bacteria. Okay, so this you will be studying over there. So this is about the photosynthetic pigments. Now once we are now we are done with the photosynthetic pigment. Now in next session we will discuss about the paper chromatography, absorption spectrum, action spectrum, all these things. Okay, so that's all for today. Thank you class. Thank you so much.